Hello everyone. So we are here today. We are going to start the biology. So in the biology, as you know, we have in unit number 1, reproduction. We have successfully completed the chapter number 1, that is asexual type of reproduction in organisms. So how the organisms reproduce by means of asexual, as you know, the budding type of reproduction and offset, runner, everything we have done in the chapter number 1. So in the chapter number 2, that is reproduction in flowering plants. The flowering plants is nothing but angiosperms. How the flowering plants reproduce by means of sexual. So the flower is a fascinating organ in plants. Why I am taking the flower in the description, why I am selecting this organ in plants, because this is the reproductive part in plant. So this flower, so this is the means of reproduction. In this there are 4 whorls. 2 are outer and 2 are inner whorls. So quickly we will see the structure of flower, so that we will study all the whorls in flower and what are these the functions of whorls, how the outer whorl essential to the flower, how the inner whorl essential to the flower in the reproduction. Okay. So here we are having the LS of flower. As I told you, a flower having 4 whorls, outer 2 and inner 2. The outermost whorl is known as calyx, and the next whorl next to the calyx comes the corolla. These are non-essential, like these are known as accessory whorls. They do not take part in reproduction, but when the flower is in bud condition, the calyx protects the flower, and the corolla having various color of the petal, it attracts the insects for the pollination. And coming to the essential whorls or next inner whorls, that is stamen and pistil. So in the stamen, here there are 2 more labelings are there. Let me write that first. So this is the anther and this is the filament, and now they both are known as androecium. The androecium is the male reproductive part of the flower. And now coming to the innermost layer or innermost whorl of the flower, that is nothing but gynoecium. The gynoecium consists of the basal swollen part, which is ovary. This is the basal part, little bit swollen. And inside the ovary there are ovules are present, and a long slender is attached to that ovary or swollen part, is known as style, and at the top of this pistil a cup shaped structure is there. That structure is known as stigma. They 3 are known as gynoecium or female reproductive part of the flower. So here we are having a complete structure of LS of flower. So these 2 are the outer whorl or accessory whorls, and these 2 are the like androecium and then gynoecium. These 2 are the essential whorls. Why it is essential? Because it takes place in the reproduction. Okay, so we'll study one by one. So we'll start with the anther first, or the andrecium first. We'll move on to the gynoecium, and like this we'll study all the parts of the flower and how it synthesizes or how it produces the microspore or the ovary or the ovule. This is how we'll go one by one. So here we are going to study one by one in detail. The first whorl what I am taking here is stamen. As I told you, a stamen consists of the anther and the filament. So this is the anther and the filament. If the stamen or andrecium, whatever you can call it as, the stamens or this filament, a long slender structure which is nothing but filament. So the filament, if it is attached to the thalamus or the receptacle, so the stamen filament, maybe it attached to the thalamus of the flower or to the petal of the flower, either this or that, whatever it may be. The filament, so this is the anther, as I told, this is the stamen as I told you, and anther and the filament. The stamen, this structure or this area is known as proximal end. So what is proximal end? So the proximal end is the area, or this is the place where it is originating. So you consider this is the thalamus or the receptacle, or it may be a petal from where it is originating. That originating place is known as proximal end. If I consider this end is known as distal end. The distal end is far away from the originating area. So the filament, and now coming to the anther, here I have drawn two lobe-like structure. What is that? Two lobe-like structure, what I told you, a lobe-like structure, yes, that is two lobes of the anther. This is one lobe and one more lobe. These two lobes get connected to the connective. Okay, these two lobes, actually they are separate with one another. A strip-like structure or strip-like tissue or a sporogenous or sterile tissue which connects these two lobes, that sterile tissue is known as connective. So here we have a complete morphological structure of stamen. So with this we will study what is there inside this anther. Morphologically we can study, yes, it is there, anyone can tell. Okay, so this is what is that, cylindrical in nature or versatile in nature or anything like that we can tell, but what is there inside the structure, or we can call it as tiers of anther. That is important to understand how the organisms or how the plants reproduce. So after this, after the anther or after the stamen, we are going to study the tiers of anther. As I told you, what is there inside the anther, to understand that it is necessary to know the layers present in the anther, the sac present in the anther, everything is important here. Tiers means, as you know, transverse section of anther. Observe this because this is new one. You have to understand how I am drawing, and in 2014 or 2016 I guess they asked to draw the tiers of anther for 5 marks. Consider this is the anther having two lobes, and this is one lobe and this is one lobe. What I am taking here? I am taking the transverse section. I am just taking a cut of the anther. This is how it looks. Okay. So, this is the outer boundary or outer wall of the anther. What is inside that? Let me draw first. Okay, we will see one by one. So, as I told you, outermost layer. The outermost layer is known as epidermis of the anther. That in the case of anther, actually it consists of 4 layers, or it is of 4 layers, and 4 circles I have drawn here. 1, 2, 3, 4. So, consider this is one lobe and this is one more. Okay. In one lobe, or you can call it as a theca or lobe, whatever it may be. So, we have a 2-lobed anther here. You can call it as dithecus, a dithecus anther, because here 2 thecas. One theca and one more theca. In one theca in one lobe, 2 circular structure I have drawn here. What actually it is? This is nothing but microsporangium. One more microsporangium and one more microsporangium. 4 microsporangiums are there. Why it is 4 microsporangium? Because we are taking the transverse section of dithecus anther, or 2-lobed anther. Okay. What is microsporangium? I will come little bit later. As I told you, it is it is having 4 layers. So, here the area, you can see this, the area. So, this area or this part, it comes the outer epidermis. So, outer epidermis will be flat cell, or it is made of flat cell compactly arranged and the function of flat or outer epidermis is protection, or it is protective layer. It protects the anther. So, that is the first layer of anther. As I told you, there are 4 layers. So, this is from here we are starting. This is the outermost layer or outer boundary. This is the first layer. Next to the next layer is, or second layer will be, this is the first layer, and second layer will be beneath to this epidermis. Let me label that. So, this is epidermis. Beneath the epidermis, radially arranged or radial cells compactly arranged endothecium cells are present, or endothecium layer is present. So, this is how it will be. And one more thing to tell, the endothecium is thickened by, or it is deposited by callus. Next, so this is the second layer. Let me label that also, endothecium. So, next to the endothecium, there is the next layer, or we can call it as the third layer. The third layer is of 2 to 4 layers, or 2 to 4 cells are present. Two to 4 layers of cells. Here the cells are loosely arranged and parenchymatous. Okay, so this layer is considered as middle layer or middle wall of the anther. So here we go, the first layer is epidermis, second layer is endothecium, and the third layer will be the middle wall or middle layer. Okay, so I didn't talk about the fourth layer. As I told you, there are fourth layer, there is no space at all to draw. Here is the microsporangium, as I told you, the wall of the microsporangium. So here, this is the fourth layer of anther, TS of anther. So the fourth layer is known as tapetum, made of sporogenous tissue. In between these two lobes, there is a strip of sterile tissue. That strip, as I told you, which holds the lobes, two lobes it holds, that tissue is known as connective. The name itself saying it connects two lobes. Okay, here we go. The four layers, the first layer is epidermis, second one is endothecium, and the middle one is the middle layer. The next three layers are known as middle wall of the anther. So this is how we'll study one by one. There are four layers, and the fourth one is the tapetum. Little bit we didn't heard about this tapetum, what actually it is, tapetum means what, we didn't heard in the first year. Okay, no problem. The tapetum is the nutritive layer. As I told you, this is the fourth layer of anther, and the tapetum is nutritive layer. Why it is nutritive? Is it supplying any nutrition to the anther? No, it is not supplying nutrition to the anther. It is supplying nutrition to the cells present inside the microsporangium. What is there inside the microsporangium? So this is these are nothing but the microspore mother cells or microspore. What I'll come to that little bit later. So the tapetal cells, one tapetal cell, one more tapetal cell. These cells provide nutrition. And one more thing, the tapetum is multinucleated. Multinucleated means more than one nucleus is present in the tapetal cell. Why so? Because here endomitosis takes place. Endomitosis means karyokinesis occurs, but after karyokinesis, cytokinesis do not occur in the cell. Because of that, multinucleus is present. So the basic concept is, or the thing is, the tapetum provides nutrition to the microspore mother cell or microspores. Now here we have a structure of TS of anther. Still we don't know where is the pollen grains. Where are the pollen grains will be. So if I draw this diagram, consider this is the young TS of anther. In the young TS of anther, we are going to call this as microsporangium. Why so? Because here still the pollen grains or still the microspores are, they start germinating. Still it is not. Still they have not germinate, or it is in a young condition, or the young anther is known as microsporangium, or this layer is known as microsporangium. What do you call this as, or when it grows or when it matures? What do you call it as? It is known as pollen sac. Remember the young anther. If you are drawing the young anther or TS of young anther, you have to mention this as microsporangium. If it matures, it becomes pollen sac. Why? Because it contains number of pollen grains. That's why. Okay. So we'll move on to the next concept. So we studied the young TS of young anther. So this is the microsporangium. what happened, how it develops, how it changes its position when it matures. I'll draw here. So I'm drawing next to it, no problem, you can follow this only. So the young tiers of anther internal structure. So this is the second stage, or when it develops, how it looks. Nothing will change. The layers will remain constant, or the layers will remain as it is. Only the microsporangium, it get little bit wider, like that. And the cells present inside the microsporangium, they will be like this, little bit spacious. Only it grows little bit spacious, you can call it as. Now we have number of circular shape structures here, inside this microsporangium. Okay, so these structures, microsporangium or circular shape structures, are nothing but microspore mother cell. So these microspore mother cell undergo microsporogenesis. So what is microsporogenesis or sporogenesis? The microspore mother cell undergo meiotic division. They undergo meiotic division means when it undergo meiotic division, you know meiotic division or meiotic stages. When it undergo meiotic division, it produces microspores. What actually produces microspores? The microspore mother cell produces microspore mother cell produces microspores through meiotic division or meiotic division. The process is known as microsporogenesis. So microsporogenesis, the process completes. It takes a long period. After the microsporogenesis, after these microspore mother cell, it forms tetrads. What is tetrad? Tetrads are nothing but having 4 spores, but you can see only 3 spores here. Where one more spore is, it will be behind to these 3 spores. So 4 spores are the resultant spores of the microsporogenesis. One microspore mother cell give rise to 4 tetrads. So after that, after some time, after some period, these tetrads, they liberate, they dissociate or they desiccate with one another and form number of microspores, or literally you can call it as pollen grains. So you can observe this here, a little bit incision is there, little bit bend is there in the anther. Why it is so? The bend, why the point is little bit bent in nature here. So this is the point of dehiscence. You know the word. What is dehiscence? Dehiscence means liberate agodu, horgade hogodu, discharge agodu anta artha. Okay, so this is the point of dehiscence. One dehiscence, one more point of dehiscence. After the maturation of the pollen grain, like first it is microsporangium and then microsporogenesis occurs. After the resultant product of the microsporogenesis is formation of tetrads. These tetrads desiccates or they dissociate and forms number of pollen grains. These pollen grains should come out from the anther. From where it will come out? From here, from the point of dehiscence, the pollen grains all will come out from the anther. So what is the use of coming out of pollen grains? The pollen grains should sit on the stigma of the female reproductive organ. Why? For the reproduction, from where the process starts. So this is the internal structure of TS of anther. Today you have studied all like the LS of flower you studied, and then how the anther it looks, and TS of anther you studied how the microsporogenesis occurs, from where pollen grains discharge. Everything you studied studied today. We will continue